Fernanda here to the front. That is one of the organizers that is joining us. And then you'll meet Mao and Bill also later today. And so, where are we? Yes, Mao is here. and Bill is on his way. So yes, looking forward for this next week. And now, Christoph, floor is yours.
Thanks so much. Okay, welcome back. Today's my last lecture here. I have to confess I really enjoyed it. Literally from morning to midnight, the entire good week. Since I was asked a few times about my lecture material, and honestly I sent it on Monday after I arrived, and it somehow didn't make it to you, so that's the feedback I got. So I felt like, and someone came up with the idea for a QR code. I might have said like, oh, why should I do this? Okay, then I did it. So if you, I hope it works. When you download it, it's a big file. It's almost 1 gigabyte. It includes all the PowerPoint presentation or keynote, and also my handwritten notes. Yeah? I mean, they're supposed for you usage to learn the material, right? So if questions pop up, I'm sure you find typos, notation inconsistencies here and there, because I was teaching this subject a few times already, but then I, of course, combine certain notes. So in case you find some discrepancy, I mean, just like be critical with reading, also minor signs, etc. Yeah? But the material is really for you, so in case you decide to publish it, include me as an author. Okay? But of course, you're invited to drop me also an email anytime in the future. Good. Today I will do a little bit of a different type of talk. I felt like I always gave a little bit of a few slides at the beginning to motivate the long calculations in general. Sorry about that. Today I felt like I should give you a little bit of a glance of what we are doing in the group at the very moment. And I used a science talk I gave on various conferences recently, and extended it a little bit. And in between I will have short intermezzos on calculations to show you certain models, approaches in more detail. That's a bit the plan for today. Yeah? And ideally, if you can follow the talk and get the messages, understand the approaches, that would be the ideal feedback for you guys. Oh, now you're ready to go, and go even to a workshop and learn more about what's going on in this field of biomolecular condensates or synthetic condensates. And even though Patricia recommended me I should do condensates on surfaces, which would be a nice fit as well to what she did, I mean, I had to prepare on the weekend, so I had to decide either I prepare a new lecture or I enjoy Sao Paulo, and I decided for the second, and I had this lecture already. But however, I mean, we also in my group, we work a lot on interaction between membranes, often, for example, simple vesicles, and biomolecular condensates. And there are a lot of new exciting phenomena have been found recently, far beyond classical wetting. There's some angle inclined, etc. So there have been people found that condensates can scissor membranes, have found anomalous shapes, which are set by continuous fluxes to the triple point, where condensates wet membranes. Beautiful set of phenomena, but in my lecture series I focused exclusively not to talk about wetting because, again, it opens up a new thing to discuss. But in case you're interested, feel free to approach me this afternoon, around lunchtime latest, because I leave in the late afternoon, or you can also drop me an email. Okay, so today I would like to show you something I find very exciting, and this is a bit motivated by a fundamental question, and this is, you see all these droplets in the cell, but it's not, first of all, we don't know what they're doing, most of them, and we want to make them dissolve at some point. So you ideally need to have a clock for these condensates to form and then to dissolve at some point. And with a chemist, your Böckhofen, we came up with the idea, well, isn't it possible that you can have condensates that form and then dissolve, and then they completely disappear, but they come again with a certain frequency, and do this all the time. And it was very funny, because like we had already a model for these active shells, remember my colloquium talk? And we're very, I'm a very lazy scientist, it seems. We just added one more reaction. Of course, I mean, you could say like, yeah, but you played hundreds. But actually it was the discussion with the experimentalist who understood the theory pretty well. And he told us, I will propose you now a set of reactions which I think can work, but I can make work. That's a very interesting guidance, because if you believe a phenomenon should work in nature, why not talking to experimentalists and they tell you a little bit what is possible, and not just making up completely independent some reaction that doesn't exist, with some reaction rate that doesn't exist in life. Good, and that's what I will talk about today. I mean, here I have one motivation slide of like really fundamental observations in the field of chemistry. I think you might, in your fundamental lectures, might have seen this very famous reaction, Belousov-Tabatinski reaction. It's a set of nonlinear reaction, nonlinear, very important, yeah, with a simple dilute diffusion term that can give rise to these spiral patterns. So this is very classics, but the idea for these spatial heterogeneous patterns to form and spirals is at least a set of equation. I think essentially you need two. I need three components, and the reaction to be highly nonlinear. Another very beautiful class are reaction diffusion systems. So this is a work from Peter Schwille, and there have been a lot of beautiful work on understanding the mechanisms by Erwin Frey, and I think he had been here a good year ago, right, in this school. Yeah? So I think you know all this now, kind of. But they form differently, because you can think about reaction diffusion equations you know already. You have a linear diffusion term, so d times Laplace of concentration. You have a set of concentration fields, typically also 3. But then you have nonlinear reactions. So the nonlinearity of the reaction drives this kind of pattern. So if they have been linearized, nothing happens. Then nothing of this will form. Today I will show you the opposite. I will show you how linear reactions can form related patterns. In that case, the formation of the dissolution of droplets. Good. Yeah, this I've shown you already. That's why it's a bit the motivation. So why condensates at all? I think this we can walk through quickly. Of course, this is kind of when you do science, you always take some inspiration, and I take a lot of my inspiration from observations from biologists. So let me give you a few examples what had been known even before we wrote down the model. There had been a beautiful work by the group of Stefan Grill and co-workers on a very classical system in the end. So classical system, I mean polymerization of actin. Yeah? It's a bit more complicated here, get a very simplified sketch. So what you see on the left-hand side are actin monomers. Yeah? And on the right-hand side, this is polymerized actin, also branched. So they form these long strings. Yeah? Actin has a persistence length of around 10 to 20 micrometer. So they're not necessarily stiff, but depends on the length scale of this illustration. And it's an out-of-equilibrium process to make these long filaments, because entropy tells you essentially, or thermodynamics tells you like, nah, it's difficult to get these long things, so you better take an out-of-equilibrium system. And it involves a lot of names and molecules. I don't want to get into this too much, but it has essentially activating units. Yeah, that kind of, this is the activator, WASP, and this one is actually the builder to make it longer. Yeah, it's an out-of-equilibrium system, so requires ATP. But there's also a counteracting process. So even this, the backward pathway is not thermal, not spontaneous. So it uses molecules like ADF, cofilin, etc. So this you can look in a homogeneous system, but they've done this at larger concentration, and then they found this very interesting phenomenon. What you see here is an overlay of a theory and an experiment. I don't go into details of the theory. It's a coarse-grained reaction model, very elegant. But what they plot here is the concentration of WASP, so this activator. So obviously, the more WASP, you have more this pathway. And the actin concentration, it just tells you you have more of these branched filaments. And what they observed is they have seen like a one-loop activation of that. It goes up here, and then it comes down here, in the theory and in the experiment. The reason for that, I mean, they have also an illustration in the paper, but sometimes it's also very nice to look at these news infused, yeah, 2S, articles, in that case by Guillaume Charasse and Martin Lenz, a very beautiful one, and I think this illustration is really explaining it very, very accurately, I find. And then what they have observed in the experiment, which is not shown here, of course, on the left, is that there is a condensate rich in WASP, so this activating unit, but also in terms of actin. And this WASP, we see illustrated this activating unit as this big sphere here, and then they can polymerize actin. They keep on doing this, so the droplet can grow, there's more actin around, etc., etc. And then at some point, the mechanism is not known. It's also written in the paper. New research, exactly, not everything is always clear when you see something very exciting. Then at some point, WASP leaves. And thus, this pathway gets more defavored, and this backward pathway essentially depolymerizes all the actin and also dissolves the droplets. And this kind of drives this one-way activation loop also here illustrated here very nicely in this sketch by Gioma and Martin. Okay, so this was actually a very interesting system, but so droplets can form and dissolve, and then they stay. When you think about this a little bit, and in the business of actin, you know, like, if you keep a little bit of actin around, yeah, there is a secondary nucleation pathway actually that you need to restart the cycle, yeah. Because in principle, when you end up here, why do you not restart the cycle? The reason is that you need secondary nucleation. When all filaments are gone, the activation can't happen. So if you keep a little bit of surface, you can see this even as a stochastic oscillator. Huh? I mean, the paper was sold for something else to provide a timing mechanism. But when you think about it, so as long as you allow it here to jump off again, the system is just oscillating. Yeah, I talked to Stefan Grill about this, a long discussion. Like, yeah, you're right, exactly. It's a stochastic oscillator. Yeah? Then there's another paper from the Helpberg and co-workers. They looked at a special protein called P53, and they saw cycles of formation and dissolutions, even in vivo. They claim, but it can be discussed, I had a nice discussion with him a few months ago, that this is kind of, these oscillations help kind of to regulate the size, and this optimizes DNA repair. Yeah, so if you want to have a look at this, whether this is convincing, I would highly recommend you to have a look at that reference. So they have a function. So you think about a functional rate of these oscillations. So this was enough for us. Then I talked to Jörg Böckhofen, but then he came with a different experimental observation. Then we worked on this one. But it also led to an oscillator, but with a different mechanism I will show today, and I will show you the following. So this is a big, what you see here in white is a big water droplet. Yeah? It contains, remember the talk I gave about the shells? You had the A and the B component, and with fuel, we were activating the formation of B. Same thing here. But there is something different, and maybe let the movie run. So you see that there's kind of continuous rain from the top, coming down. No, not really continuous. It comes in waves, right? See that? And below, there's something like, I don't want to call this, maybe like a mother drop, kind of, that is growing, but also, well, the system runs forever like that. So what's going on here? We understood this now pretty well. We also wrote a paper on this. Actually, what happens is you have nucleation at the very top of the container. Yeah? Droplets grow, they take up material. When they're too big, they're heavier, so they're sediment. It's just because of gravitation, simple as it is. Then they come down here, and then they fuse with this bigger droplet. And at the equilibrium, the system would then stop, because then just the big droplet sitting at the bottom. Yeah? But the system starts again. And how can it start again? Because the system has a stationary radius. Remember what we talked in the last lecture, right? There was a stationary radius, an optimal size. And by all these fusion events, we could show that you are above this optimal size. So what does the system then do? It will shrink. It will shrink and expel material into the bulk. And then diffusion brings it up here to nucleate at the very top. Why denucleate at the top? This is a bit a chemical detail, because they are charged at the boundary, and nucleation is not just a matter of concentrations, but also a matter of the local electrostatic conditions. So this keeps on going forever. Yeah? And this is simply because it's an active droplet that you push above the stationary radius, and then it shrinks again. And then that's why it starts the cycle anew. We could put this into models. I mean, we did multiple approaches, but this here, what you see here, relies also on the description you have seen with these. When you have many droplets, you write down a coarse-grained description for radii, Ri, position, etc. Yeah? You can add gravitation for the center, very easy. For the larger Y equation, you have a bit of jiggling. Yeah? And what you see here, just to explain it, I mean, we have here a bottom layer. We just blew up the system. This is what we can do with the theorists. We can play with different geometries and lots of kind of things. And modeling this round boundary is not, I think, a good idea in general, because droplets can hit here this round boundary. They do slip. Come on, this becomes modeling work. So we felt like, okay, come on, let's make it simple. Let's make a box that is here. It's a closed box at the bottom. At the top, you have periodic boundary conditions on the right and left, and we can play with the dimension of the height, etc. Yeah? It comes also with a nice advantage, with much easier eigenfunctions. You have then a flat layer here at the bottom almost. Okay? So when you do a zoom in here, you see droplets sedimenting. You see, like, the layer has a heterogeneity, but also droplets are heterogeneous. Yeah? And you can see this with this beamer, I think, right? And this comes exactly from the fact that we exactly solve the eigenfunctions inside and outside, and that's important because you need the gradients inside and outside to have continuous fluxes through the interface. Yeah, and let the movie run. We see the same thing. You see these waves coming down. Then the layer increases here. This is here. And then it goes up because they all come down. And then it shrinks. It's above the stationary size. It shrinks and nucleated more the droplets at the top. And this also a theory. Lucky us, it keeps on running forever. Okay. So the interesting bit is everything what you have learned up to now, in principle, we could do as a project work tomorrow, because you have all the concepts ready. So little tricks here and there. One has to learn addition, but then to implement something like this and compare it by the numbers with the experiment, you would be already in almost the position to do it. Okay, this was I forgot left over. So, but this kind of oscillation needs gravitation to bring the droplets down, right? And then you're above the station radius, and then you keep let the cycle running. We felt like, is it possible maybe that a single droplet can already oscillate? Yeah? And here, this movie I've shown already to you. This is without external driving, so no one oscillates anything in that system. The droplet is kind of a self-oscillating droplet. Yeah? It forms and dissolves and keeps on doing this forever. Yeah? And today I will explain you how to do this, what concepts are involved. So I will show a few things on the chalkboard, but then explain you essentially kind of the links to reaction fusion systems, nonlinear dynamics, kind of to link it up with concepts you've already learned in the past. Good. So what's a minimum model? So let's stay simple as we can. We need a solvent. Chemistry occurs in a solvent. We need a component that phase separates from the solvent. We call it A. And an addition component B that is the product of this reaction. So that's the reaction we talk about. We call this reaction R1, already indicating there might be a second reaction we have to talk about today. So if I have such a system at equilibrium, remember what I told you. When you have an equilibrium system, then you go to chem equilibrium, and also phase equilibrium, the combined one. And that's just a single, so the thermodynamic phase diagram of such a system, it's a ternary system. You can plot your temperature or some chi parameter, and then the other axis is set by the conserved quantity of the system. The conserved material in a reaction between A and B is simply material A plus B. Okay, that one half is not relevant. And that's how the phase diagram would look like if we have an upper critical solution temperature, okay? And we obey the equilibrium conditions you've already learned, and this is phase equilibrium for all the components I and chem equilibrium, okay? Good. But we want to have something out of equilibrium, and a self-oscillating droplet that oscillates forever cannot be at thermal equilibrium. So what we have to do in a nutshell, as I alluded already a few times, we have to open up the system. So we have to exchange mass and energy through the surrounding. And I stay with this simple theme of fuel and waste that you've already learned. But of course, one can add, you could also think about exchanging the solvent in principle. You could do evaporation cycles if you want. There's lots of possibilities. Okay, but we stay with this classic scheme you've already seen. And an active droplet is exactly the one where you have, at a steady state, a balance between the in and the efflux through the droplet interface. Good. Is a single reaction enough for oscillations? And if I ask that question, obviously the answer should be no. But let's see what we have to add to the system to make it oscillate. What are the physical equations that we want to solve? Of course, you can start with this Cahn-Hilliard field transport type of equations. So where we define here one fraction field for each component I as a function of space and time. We have this conserved part that is simply the divergence of a flux, and the reaction part. This you have seen. The flux, we've also seen that one is driven by the gradient of the Kemp potential, and it gets multiplied by kinetic coefficient that tells you how fast you diffuse. Again, diffusion, please, latest after this lecture, diffusion is not driven by concentration gradient, it's driven by gradients in chemical potential, always. The Fick's law is a special limit for the dilute systems. And the K potential is again the functional derivative. This we already see. Like you know all this now. That's good. So the reaction here, to see something interesting, we have to of course break the balance of the rates, and I do it as follows. I come up with two rate coefficients K1 and K2, and allow myself to have here an extra K potential, and this is exactly the K potential I introduced in the lecture. This is this K potential difference between the fuel, or can be, for example, the K potential difference between the fuel and the waste. Good. Yeah, but of course, when you do science and want to get at some aim, like oscillating droplets, you need a freedom to play. Yeah? And the freedom to play here is what we call the fueling function. Yeah? So we allowed ourselves to come up with a funky form, okay, and explain step by step why we choose like this, and whether it can also be realized experimentally. Yeah? But of course, we have a certain freedom. So what we have to do essentially, in a nutshell, to break the balance of the rates, we have to make this extra chemical potential contribution here state dependent. Okay, that is different between the inside and the outside. Then you break the balance. Then you can't rescue thermodynamics. Yeah? And how to do this? Yeah, I mean, the reason why we did this one, choose this tan H function here, is simply it allows us to have a sharp transition when we have a lot of material. Phi, so the psi is phi A plus phi B. Yeah? So if you're above this threshold, you switch something on, while below you're off. Okay, that was a little bit the idea, to have something like a switch to be active, while at too low psi values, so this is a conserved quantity A plus B, you don't fuel. Yeah? So in a nutshell, what the experimentalists have to do, yeah, what is the rule? Okay, that's a conserved field, I've mentioned this already. So what's the recipe? The recipe for the experimentalist is exactly this one. So we need a droplet where B phase is better than A, and the dense phase is fueled. Yeah? That's what the experimentalists have to do. So they have to make sure that the fuel is hydrolyzed in the dense phase. Okay, so you Pukov was sitting down and said, okay, how do I have to design on my fuel that it goes in dense phase, gets hydrolyzed there? That's the rule. Good, so that's how it looks like. So what I draw here is a ternary phase diagram. So you see here these two volume fractions, B and A. So this black line we have already learned. It's just why we choose that one, it's a bit magic. It has to do exactly with this criteria. You cannot appreciate up to now. Why should B phase rate better than A? Well, we played. If that is not, if it's the other way around, there are no oscillations. This I can tell you right away. If you implement this idea, B phase separates better than A, you get such a phase diagram. Okay, what do you see here? So B can phase separate. You see this? Here, that's B. It's connected to the axis, so that's phase separation. A can also phase separate at this axis, but you see, like it happens at much larger concentration here versus that one, yeah, and it has a much smaller domain. It's exactly implemented in this diagram. Okay. What's this funny color code here? This is exactly this fueling function. So you see, like how we choose it. We can choose it by choosing this this psi delta. Yeah, here everything is fueled. So this entire branch, that's the inside branch, is fueled, while the outside is not. So we can choose it a bit. We can move it around. It's very robust. As long as we don't start fueling the outside, then the phenomena are different. Okay. Yeah, we can write down the chemical flux. This is the well-known form you would do according to theories from Kramers and others. So we have a forward flux driven here by chem potential of mu A and mu B, and if delta is 0, if mu A and B is 0, then we are at chemical equilibrium. And we have, of course, here a reaction rate coefficient that tells us how fast the reaction reacts. Okay? This is, of course, what we extract also from experiments, because we need to know how fast the reactions are. So it's okay, the value of K1 is very relevant to see what we see later on. Good. So that breaks the balance of rates. This you've learned already. But as I told you, I mean, the problem with these Cahn-Hilliard type of equations is you push a button, you make this nice movie you've seen, but you don't go anywhere. Yeah, so I will now show you a new approach. So what you've seen up to now are narrow interface models we discussed. Yeah, so we have a boundary condition at the interface, and we look for the eigenfunction, but we rely on an assumption that is maybe true, maybe not, and this is this quasi-static assumption. Yeah, otherwise, a time-dependent reaction diffusion problem is hard to solve. I will now show you a different framework, and I will derive it from the Cahn-Hilliard equation for you on the board. And this is so much simpler. And what is it? So what do you see here? So first, you should appreciate before we had a PDE, a partial differential equation. Okay? So you have a discretized space, and what you see here now is an ODE, right? So the dot here is a total time derivative. Here's also total time derivative. What are the contributions here? So what is this phi I? Phi I is simply one fraction of component I in the respective phases 1 and 2. We have two phase coexistence, so we have two phases, yeah? So now, but now I'm just interested in kind of a homogeneous one fraction field. And it carries a few interesting terms that you would expect. It has a reaction rate, R1. It has a flux between the phases, J1 and J2. Here it goes inside and outside. And it has a contribution when the droplets are growing or shrinking, okay? Moreover, we can write down also the what determines droplet growth and shrinkage, and this is simply the difference between the reaction in the respective phases and the exchange from through diffusion. This is a framework I will now show on the board, how to derive it. And it will lead to something I find still very exciting, and we use in a lot of our current scientific work, and it's simply you can, with this approach, we can draw chemical trajectories that are out of equilibrium, but these are balanced broken chemical reactions, in phase diagrams. And that's something you feel like, whoa, if you would have told me this 10 years ago, it's like, nah, something weird, can't do it. A trajectory in a phase diagram, come on, this is a bit like inconsistent, but it just relies on the idea that there can be situations where we have two equilibria here, chemical equilibria, and which we don't want to reach, right, because we break the balance of the rates, and we can have phase equilibrium. But we can have an out of equilibrium kinetics where the system is always at phase equilibrium, and this is when the exchange of the phases is fast enough, and that relies on diffusion. So you could ask, like, yeah, is this a realistic thing in general for these condensates? I can tell you right away when you plug in real numbers, I would say it's the rather typical case. And this is something we develop in this lecture a little bit, and I show you now on the board. Good. So that's why I call this, I just call it here, a very rough, fast diffusion model, right? And let us derive it systematically. I mean, a few things I would like to specify just for the sake of presentation. I would like to look at a ternary mixture, so we have two independent volume fraction fields, and of course the third one, say it's a solvent, we can just calculate by 1 minus phi A minus phi B, and as I said, this is not incompressibility, this is always true. So, we look at a single chemical reaction for now. Later we also introduce a second one, and this is simply A rect to B. And we start with a continuum theory. You know how to do it now, right? Use linear Onsager, where each one fraction is a field that depends on position x and time t. And I is, of course, an index of the two independent components, A and B. The transport equations of this continuum theory is very simple. I know I'm a bit repetitive now with what I've already put here. So I can write down partial differential equations for each field. Each field gets transported with a flux. I call this JA here. And I have a reaction, a local sink and source term, SA and SB. And I would like to study an incompressible system. You have learned this is that the molecular volume is constant for both. To be consistent, is of course that the chemical reaction to be volume conserving. Okay, as I said, it's not always true. The way out is to use the Gibbs ensemble, but to keep it a bit simple, chemical reactions or have to be, otherwise I'm violating compressibility. Good. That implies right away for the volume fraction fluxes that SA is minus SB, and we just call this S for simplicity. Okay. the fluxes that we have here in the continuum theory is we write with linear response. And without any cross couplings, so the flux is some lambda I that depends on, of course, the volume fractions in general, time, sorry, I'm just jumping to here to the new line, and it's driven by the chemical exchange chem potentials, rebar. For the experts in the room, there can be also cross couplings. So the flux of I can be also driven by the gradient of the chem potential of J, of the other component. This one can include, the derivation is almost the same. Good. So what do we want to calculate now? I would like to introduce, maybe write first of all the field I'm introducing. I introduce now a new field, phi I, alpha. What is alpha? Alpha is, for two-phase coexistence, carries these phase indices, right? The phase 1 and 2 is here, this nice picture on the top right. And this is it is no more a field. It's a function of time, okay? And how do I define it? Define it as the integral over space of the volume V alpha of our field. Okay, so this is the field that depends on space and time. So I just integrate this out. So I lose the spatial degrees of freedom. And to avoid that it scales with the volume, I divide here through E alpha of t. Okay? And the framework, as you can see here, is exactly the time evolution of this averaged concentration field. And we would like to understand in what limit it is fine to reduce, it's an enormous reduction, these partial differential equations to a set of ordinary differential equations for that field. Okay, let's do it. Let's calculate it. So, yeah. So what depends here on time? So, what depends on time here? The field here depends on time, fine. Yeah, what else? Where are the time dependencies here? The volume depends on time, right? Oh, that looks like an ugly chain rule with 3 contributions, yeah. Yeah, but we have to do it. We have 3 contributions. So we have to calculate the time derivative of first of this one. That's rather easy, okay? But then we have an integral over a moving boundary, okay? This calls for, among others, for the Reynolds theorem. Have you heard of this in hydrodynamics? Yeah, exactly. So we have to also include this contribution. Actually, but this one will be neglected because it just gives a surface term, but let's do it step by step. Okay. Okay, let's write it here maybe again. What is a superscript? I always do. So, what are the contributions? So the first contribution, let's write this down, is let's do the derivative of this one here. So we can just keep this. Yeah. So what we get is essentially a minus sign, 1 over V alpha of t squared. The chain rule derivative is this one here. And then what should we just multiply the integral with it? Yeah? Okay, that's one contribution. Yeah. Then there is another contribution where the total derivative acts on the field. So I can write here plus. I keep this one here. So, and I get then a partial derivative of phi I. Good. What else? I have a term through the surface. Yeah? And if you write it, you have to be careful because I did it separately because actually it drops out. Gives you a surface contribution through the boundary of V alpha. That's called D alpha. N is the normal vector of the domain. The local interface velocity, this we had already. Times the field. You can do here, that's a scalar product, okay? Hope I didn't forget because I'm... So, let's simplify this step by step. This one looks pretty good, except we borrow here 1 over volume to use the definition here of our reduced field, okay? So what is left is then 1 over V alpha, so just one volume, dt V alpha of t times, how did I call this, phi I alpha. That only depends on t. Well, that looks like a nice contribution. Huh? And if you look up into the equation, that's the one on the very right-hand side. So let's continue here. What do you do with that one? Okay, sorry, I have to think. Good. So what do you do with this one? What do we do with the partial derivative of this field? Huh? Yeah, exactly. Yeah, we apply exactly what we have. We have an evolution equation for that, which is minus divergence of Ji plus Si. Okay? Okay, so we can plug this in. So we get first of all here a minus 1 over V alpha. So you see right away, this is the volume integral of the divergence of the flux. That calls for what theorem? Gauss theorem, right? Just call it in Gauss theorem. Okay, you can do this now. Just plug this in. The minus sign, we have a surface integral, the normal vector projected on Ji. This is this one. Then let's keep on going. We alpha of t. The Si is just an integral over this local source term. So I integrate in the respective domain each local chemical source, and I get a net source for the domains. V alpha of t, Si. Okay? Then what about our Reynolds term? Well, we can keep it, by the way. I have to confess, when we derived this for the first time, we didn't write it in the science in this paper. But of course, it is a term that scales with the interface. Yeah? So then I have to, you could argue, okay, here is also an interface term, but this will be a net flux term. It's actually a bulk term that accounts for all the net diffusion events from the total bulk inside and outside. It's originated from a bulk term. Well, the other one is actually a net interface term. So when you go into the thermodynamic limit, we can neglect this term. But of course you can keep it. It's no problem. I mean, it gives you just a modified speed of your volume growth. Yeah? But we don't have to, when the droplets are big enough, we don't have to keep it. Good, and then I think we're done, because this one here, I have to look how it did do this. Okay, no, the entire thing. Okay, good. We define now as a net flux, capital J. This one here, did I define right? Okay, is a net reaction here. Sorry, I have a clash of notation, I just realized. Okay, let's call it Ri here if we want. And this term we have already. So you nicely see that you get these reduced set of equations. But deriving is, or integrating out to special degrees of freedom doesn't mean then you have a closed set of equations. So what is the condition physically for this to give you a closed set of equations? For that, we have to remind ourselves about the reaction diffusion length scale. Remember, when we have a first order rate, the first order rate means when you linearize chemical reaction, that is the kinetic prefactor of a chemical reaction. Yeah? K alpha. So in terms of units, it's 1 over time. Okay. Be careful, because chemical reaction rate coefficients can have, depends on the reaction order, can have also units 1 over time and multiplied by concentrations. The reaction diffusion length scale that is essentially built by the square root of what do we have? We had this already, the diffusivities in the respective phases divided by K alpha. So we can build in general multiple reaction diffusion length scale because we have different domains. But it tells us once we are below, so say we have a box, it has a condensate, for example, 1 and 2. We have multiple length scales here in this problem. We have, for example, here radius r. We have here system size L. Okay? And if, and that's exactly the validity of this framework, of course, obviously the radius is smaller than the system size. And as long as the reaction diffusion length scale, need not to be much smaller, but at least, that's fine, it's fine like this. As long as we have such a hierarchy that the reaction diffusion length scale is bigger than the system size, or if there are multiple droplets, bigger than the interdroplet distance, then we know that diffusion dominates in each of the spatial domains of our problem. Okay? Which implies two very important things. Okay, wait before I say that. Okay. Exactly. Okay, this we define as fast diffusion limit. This gives this framework also the name. Okay. So let us write down the flux as we had in the continuum theory to understand what this limit means. The flux in each phase is exactly this one here. So the flux is a physical quantity. It tells you how many molecules go from one phase to the other. So whatever limit you want to do, this physical quantity has to saturate at some point. Even though you play with the parameters, you play with the diffusivity, you play with the reaction rate coefficient. So if you want to do a plot, and plot say J A, the amplitude of JA or of JB, as a function of this reaction diffusion length scale, you would expect that for large reaction diffusion length scale, you see some sort of saturation. So whatever it does, because you have gradients, etc., but at some point you saturate. How do you perform this limit? How do you perform this limit? Fast diffusion means that the kinetic coefficient goes up, and thus the diffusivity goes up. Okay? Moreover, what happens, so this one goes up, this one goes to infinity, if you like, okay, this part here. But what goes to 0 is the chem potential gradient. Okay? Such that the flux becomes a finite value. Yeah, I mean, remember, always when you think about some limit, say you start describing diffusion on a lattice, yeah, hopping process, yeah, you introduce, you want to do the hopping rate to infinity, but also the distance of the hopping to 0. And then you have to think about, okay, but what quantity do I keep fixed at that limit? And the quantity that is fixed, it tells you how fast you hop, and that's the diffusivity in the end. Yeah? Same thing here. Yeah? So the transport gets faster and faster, large diffusivity, but then this product becomes finite, or remains finite. So what do the two conditions imply then? If diffusion is fast, it means that phases become homogeneous. Even though they're chemical reactions. So think about, I convert the molecule A to B. I've locally created a concentration perturbation, but then diffusion is so fast, it immediately gets homogenized. And it also equilibrates between the inside and the outside, because, and how do we see that? Because the gradient of the chemical potential goes to 0, which means we have a chemical kinetics at phase equilibrium for all times. So at each time point, even though there are out-of-equilibrium chemical reactions, the system is at phase equilibrium, and you know how to write this. This is simply mu I1 is equal to mu I2. So what do we have then? We have in the end kinetics. So this condition, this is an algebraic condition. That is the condition that calls for the binodal line. So which means this framework describes a chemical kinetics where you move along the binodal manifold. So whatever phase diagram you can imagine, whatever dimension, you have always a binodal somewhere. It's a surface, a high-dimensional surface. But this kinetics is just moving the system along the surface. Yeah? And you could argue, yeah, it's just an approximation. Fine, it's an approximation, true. So you can deviate maybe a little bit from that surface, but it's the guiding manifold for your kinetics. So movement. And the right here manifold, because it need not to be a line. I mean, the phase diagram we had in the previous slide, I will show it again in a few minutes, was a line, but in general it can be any, say the phase diagram has d dimensions, it can be the d-1 dimensional manifold. So, why is this framework interesting? First of all, it's simpler, because instead of solving a PDE, you solve a set of ODEs, okay? That's one good reason. Moreover, we lose, in the coarse graining, we lose parameters. So we lose all the mobilities. We have less parameters, which makes sense, because the final movement should not be dictated by the diffusivity values. Exactly, and that's why it becomes very powerful. And finally, and this is exactly what I was showing now step by step in the talk, you can now, because we understand phase diagrams, right? We understand phase diagrams because, yeah, if you open up here, then the chi parameters are different, you like each other more or not. And now you have chemical kinetic trajectories on the phase diagram. So if it goes in one domain, we can understand why. Yeah, that's where it gives you a way to understand this very complex, this set of very complex systems. Yeah? Good. So, but we can also write it even more simpler. So if I have now homogeneous, not fields, but functions in each of the domains, I can also introduce so-called average quantities, yeah, which are more reflecting the actual chemical nature. So we had this A to B reaction, remember? Yeah? The A to B reaction comes with a conserved quantity. The reaction can't change. It's simply A plus B. Okay? However, what is actually describing the progress of the reaction is the so-called reaction extent. A few students asked me about this in the last lecture, which is essentially the difference. Okay? And why I'm making a big fuss about it? Because here, space is still encoded in these superscripts 1 and 2. Okay? But now I can even rewrite this in terms of these averages, and then I'm just left with 2. With 2 scalar fields to describe this very complex spatial heterogeneous system. Yeah? And this will be very important for the talk because I will show you how to derive normal forms. Normal forms to describe the onset of Hopf bifurcation that is involved in the system. Good. How does this dynamic equation look like for this AB reaction? That is simple, I would argue, right? So the time evolution equation for this conserved quantity for this AB reaction is trivial because it's just, where's my, here it is. It's just 0. But for the reaction extent, it is just R1 bar, one can show. A bar is just the average. It's just always volume averages. So what is the bar quantity? So to get dot, I mean, it's very easy. So we just multiply whatever quantity it is in that phase times the volume plus the other one and divide through the total volume. Yeah? This is what the bar quantity is. And thus, if you know the reaction, so R here, you see, this one we know in each phase. So you can calculate the R bar simply by that one. So we know it. Very easy. Yeah? So it's a single scalar ODE we have to solve for that problem. It gives rise to a concept that is called the reaction milk line. That's simply the line where the reaction does not progress. How do I see this? Because the dt of this reaction extent that tells me about the progress of the chemical reaction is 0. Okay, it's a special stationary state, and it will be important for the next slides. Okay. So that's the phase diagram I want to look at. I motivate a little bit why I want to look at this. Otherwise, you don't see the oscillations. B here on the y-axis is phase separating better than the A. Okay. The black line you see is the binodal line. You know this. This domain is just the forbidden domain because then you would have volume fractions when you add them all up above 1. Yeah, that's why there's a triangle. Remember Patricia's diagram, they're more elegant, but for me it's too hard in my brain. You just can do a triangle with equal side length and to illustrate the same thing. Yeah? And when you don't do this, you get this gray domain on the top right. Good, that's the reaction we talked about a few times already. And what is this line? This is the conserved line. Yeah? Say you're an experimentalist, you add a certain amount, you start the experiment, you start with a certain amount of, I don't care, let's say A, yeah? But no B, you sit at that point. And then the average concentration in the system have to stay on that line. Okay, you can't deviate from that because A plus B cannot be changed by the reaction. Good. Yeah, and that's exactly introduce a little bit kind of the next part I would like to do on the board. And this is I want to explain in a very nice concept. And this is a line that actually selects non-equilibrium steady states by just plotting the phase diagram. Yeah? And for that, I would like to maybe just clean the board quickly. Yeah? And now this is getting more and more to the relaxed parts. So I will now do a lot of drawings to illustrate to you the concepts. And I promise you we end up the lecture with a lot of nice movies as well. Yeah? But yeah. Let me clean this first. I was told there's even a bigger chalkboard somewhere.
Excuse me,
you want to
Professor?
ask a question? Yeah, please.
Yeah, I have a question. About this last part, you said the chemical concentration drives the binodal line. Can you explain with the diagram?
This one?
Yeah. Thank you.
Yeah, there's no chemical reaction up to now. That's not yet. This is exactly what I want to draw on the board. Yeah. So this is first of all just see it as a phase diagram. Yeah. And they say I have some chemical reaction, then the averages can't go away from this line. That's the only thing you have to understand up to now. Yeah. And why is this? Because phi A plus phi B is constant, and that corresponds exactly to this line here. This is exactly this one. But I draw it step by step. Yeah? Just stay tuned. Not yet reactions. Yeah? Okay. So let's do a few drawings. I call this part the tie line selecting curve. Okay, so let's start first with simple, with chemical equilibrium. So we can discuss this context here with chemical equilibrium. We have a system. Again, we look at this ternary mixture that I also illustrate here. I have a system that can relax. So system later we don't allow this, can relax to equilibrium. When I just write equilibrium, I literally mean thermodynamic equilibrium in that case, because we have two equilibria. This is phase equilibrium plus chem equilibrium. Okay? And the reaction, of course, that we stay with the simple reaction, okay? That there's no confusion up to now. So chem equilibrium means we have here the chem potentials between the two components are the same, between the two states. And phase equilibrium is that for each component in the respective phases, we have just two phases. They're the same. And I is A comma B. The C component we don't have to write because this is constrained by the others. So for these systems, we can make two interesting drawings. Yeah. For simplicity and also that you learn a bit the transfer, I changed the binodal shape. Yeah? So don't get confused about that. Because that's exactly the freedom, and that's a bit special about that project to get the oscillations. But happy I had a B at least, yeah, fine. So now make a slightly different binodal. I can draw something like this. The reason is this one is a bit hard to draw visually on the board. Binodal. This is, by the way, also a very interesting binodal because, remember this example of ouzo? You have oil, say A is some oil, right? You have oil and C, the C component is water. So you have phase separation of the oil. And if you add the alcohol, you bring the oil into solution. So then this component would be the regulating component that kills phase separation. Okay? This is the binodal, and it comes with these lines. Yeah? Can someone just call in? What are those lines again? How they're called? Do you remember? What are these lines called? Just say. Tie lines? Exactly. Perfect. Tie lines. Okay. They connect the respective phases. In that case, in a ternary phase diagram, giving you these equilibrium concentrations of the two respective components. So, phase separation, trivially, this is what you have learned, is exactly happening in this anti-domain here. Sorry, I'm just, on my iPad it looks a bit better. So, it happens in all this domain. So why I'm making a big thing out of it? Because this is without chemical reactions. This is just phase separation, but just for ternary mixture. And I think I just draw it because you might have not, we have not discussed ternary mixtures so much in detail with the different shapes that can exist, etc. Now let's do it with chemical reactions. Something very surprising happens. So again, we write here phi B and phi A. So we draw the same binodal. Okay, we don't want to be biased here. So something like this. Huh? This is our binodal. Yeah. But now we have also chemical equilibrium. We have a chemical reaction, so we can relax. So this one has to be fulfilled among the states. So let me have another color here. So how does that look like? Mass action law. When you super dilute here, somewhere here, it's a linear relationship. The slope is exactly this capital K constant reaction rate coefficient. Yeah? So here, we start something like this. What you might have not seen up to now is how it looks when you become non-dilute. So then it somehow, remember there was this reaction, this activity coefficient, gamma it introduced. It will eat up all these chi parameters and will make a funny shape. Okay, here it's linear, and then actually for that phase diagram, I know how it looks like. You can actually compute it. This is chemical equilibrium. So here it's actually linear. This is dilute limit. This everyone knows. But because of the interactions, it bends in general. It's non-linear. It's a non-linear relationship. Why it bends like that and not up depends exactly on that shape. You can now plot it for each phase diagram. Well, here there's no kink, by the way. It's just still here, it's smooth. Everything is okay. Good. Interestingly, now let's draw the lines, the tie lines. I can draw a few tie lines. Here's a tie line. And here's a special tie line for that system. It actually connects exactly, and it has to. The points where this line, by the way, we gave it a name, because this is called binodal, we called it mononodal. And when I say we, actually this was really new research. I was a bit shocked. So this mononodal actually exactly is connected when it comes into the phase diagram and when it leaves by a tie line. This is absolutely important, very important. We understand right away later why, but I can say it very short, otherwise the system can't be at equilibrium. And both chemical and phase equilibria are governed by when you relax back to some equilibrium, by the same free energy. Okay. So you see, at equilibrium, what you do, we're missing something, right? We have a constraint. We have a conservation law. And this is phi A plus phi B bars is constant. I call this later, I call this here, I think with a half, right? I think with a half, I call this psi. Okay? Well, how does this line look like? Because you just asked me, this is exactly this line here. Along this line, when you calculate A plus B is exactly, phi A plus phi B is exactly conserved. And this is what you do as an experimentalist. You have to select that line first. Yeah, it depends how many molecules you've added into the box. And now something exciting happens. So you can now draw all these lines here, how you can get started. There are two very distinct conservation lines. And one goes like this. And this goes exactly through this intersection. And the other one goes like this. Okay? Okay. If it doesn't look like, they should be parallel. Okay? So these are the conservation lines here. I now draw them in yellow, and I'll tell you right away why. Because before, the domain of phase separation was exactly within the binodal line. Now it's different. Now the domain where you will see phase separation at thermal equilibrium, now I'm ruining maybe my entire figure, has a completely different shape. It is this intersection cone that is set by these two equilibrium solutions. While outside, you're not phase separated. But that's strange. That's really strange. But why is this? Because when I sit here, okay, I'm phase separated at a certain time point. Yeah? But then that is my equilibrium. This would be my equilibrium state for that conservation law, but the equilibrium state for a conserved line like this is exactly that one. This is a single solution. And when it starts, you're able to phase separate when you obey both constraints concomitantly at the same time. And this is, you have to obey chemical equilibrium and phase equilibrium. And the only solutions you can find is when you start here, you will end up somewhere here, and then you end up here and in the phase separated system. It's quite interesting. So you have a completely, you see, like it changes completely the topology of where you will find on long time scales. And I just discussed equilibrium, but you will see only then you can fulfill these constraints. You find a solution because if you have decided to be on a certain conserved line, you made a decision, the system can't go away from that. And the only solution it has to become stationary is this one here. Say it's along that conserved line, it will be this one, it phase separates at equilibrium. If you have selected this one, the only solution it finds is that. You see, this is just graphical construction, but equilibrium. And now it comes. It's like, yeah, fine. Who cares about thermal equilibrium? The interesting bit comes when we break the balance of the rates. I could do the same thing. Why? Because of this long derivation here, because when diffusion is fast, I know that the system goes from one phase of equilibrium to the next. It just means it goes from one tie line to the next, and the chemical reaction just drives it there. It doesn't matter whether the reaction kinetics will bring it to chemical equilibrium or to some out of equilibrium steady state. So we can do let's draw the same phase diagram. And this really allows you to literally graphically construct exciting chemical reactions to see new phenomena. Yeah? So this again, or should be the binodal that we had already. And now I just construct a new line that is not chemical equilibrium, okay? And this line needs to be a stationary state, and this is exactly what I called before the reaction null line. So the reaction null line is a stationary state, and we have not talked about the details of the chemical reactions of this equation. So it gives you also in that diagram, it's an algebraic constraint, right? Because there's no ODE left, I just put this to 0. Put this to 0. It's an algebraic constraint with lots of nonlinearities. It gives you just a line in that diagram, but that has in general not this shape of the blue line. There should only be one unique one. And now you can play. You can say like, oh, I have this fuel, that waste, this open boundary. In general, when you're creative, just graphically you can be super creative, but then you have to talk to the chemist and say like, what shape can we do? Yeah. But I would say here, just drawing some funny lines, I would do first and then ask the chemist, can we realize this? I don't know whether I should do it blue now, but I write here reaction null line, and in a way, this one is also a special reaction null line, the equilibrium reaction null line. Yeah, let's be creative. We have to start somewhere here, and then, I don't know. I can do it, yeah? And indeed, by the way, chemically we can even realize this. And I draw this because it's a very interesting one. Let's draw here, for example, some psi to some number, okay? So what do we have then? We have, oh, awesome, we have 3 solutions. Okay, that's why I was drawing it like this. And actually one can show that this will be always a stable solution, stable fixed point, and this is an unstable one, which means we have here an unstable branch connecting. Okay, immediately calls like, I've seen this in bifurcation theory. But now we have a spatially heterogeneous system where this one selects a tie line. So there's a tie line selected. Or maybe you need a color here. What is, okay, let's use yellow here. It's inconsistent with this one, but just to illustrate here, this tie line. So this one is here, the tie line. Here's another tie line selected. There's another one, but this is an unstable state. Yeah, kind of. Here's another tie line selected. So we see that the null line in this fast equilibrium theory selects special tie lines. In that case, it selects 2. And 2 means, in the drawing, this is a bistable system. And of course, I'm drawing it because oscillations and the Hopf bifurcation underlying the oscillations will rely on the fact there's some bistability in general. No? But you see, here you can be scientifically be very creative with just drawing lines, getting multiple solutions, and asking you later, how many reactions do I need to realize this? So this is a very simple way to, and the phase diagram, you know simply from the type of molecules you have in the box. No? Good. Do you have questions on this? Yeah, exactly. Yeah. Mm-hmm.
So why can you draw the monodal line that way?
Here at equilibrium, this one?
Yeah.
Yeah. Okay, this is a bit like, there you have to trust me. So if you draw such a phase diagram of that shape, the monodal looks like that. The monodal can also bend up. That depends exactly on the sign of this chi parameter. But then the phase diagram is a different one. Yeah? But in the end, what you can do is you can think about, okay, what kind of phase diagrams can I draw in general? Internal phase diagrams, you cannot draw so many. You can have something like this I've drawn already. You can draw something like closed loop phase diagrams. And the classics one that is very common is this one here. And then you can think about, okay, how does the monodal look like in general at equilibrium? There's only one, because it's one free energy. It depends, it determines both, the mononodal and the binodal, okay? So I could draw here inconsistent 2 lines that are wrong. Yeah, that's of course, and of course, analytically, by the way, it's very difficult to solve those because it's all transcendent equations. Yeah? So you can do perturbation theory a little bit, but essentially you solve it on the computer.
Okay.
Can you explain again why, if I pick something inside the binodal with chemical reaction, but like in the triangle there, we have like but inside we have the homogeneous instead of phase separation, please.
Exactly. First of all, here, yeah, when you sit here, for example, outside, you're homogeneous, right? And if you have fixed a certain conservation law, there is no intersect with any sort of stationary solution except the one that is phase separated. Okay? Here, it's a similar situation. So the only state that is a solution to the problem, yeah, sits exactly outside. So you can be phase separated, but the system will keep on moving. I mean, this is a bit like, well, you do not. So what the system will do is actually the average will move along this line because this is the attractive fixed point. Yeah? That's same as you stay here, you would move along this line. I think this is simply it. This is a bit like when you just see it from the perspective of having an average quantity, yeah? Which you can do because any of these solutions here are either globally homogeneous, that's the one here, or are homogeneous in each phase. Yeah? And then you just ask the one, okay, where is my stable fixed point? And you just move with the average. Yeah? And, but it also makes sense because there should not be 2 stationary states of minimum free energy, right? There's either one or the other. Yeah, exactly. Hope this helped a little bit. One more, yeah.
One more, yeah. Can you explain me again the bistable system? Like, why?
Why it is bistable, or why I draw this line?
Uh, Actually, why did you draw this line in the first place? Yeah.
I mean, as I said, I mean, like I drew it just to illustrate the concept. Yeah? I mean, you have, this looks a bit similar as the chemical equilibrium line that I was drawing here. It's just a line somewhere moving somewhere. But that this one can move back, you need feedback. You need reactions that see the phase and have some nonlinear feedback. And then you can construct this. So long story short, you can also do it the other way around and say like, how I write down more complicated chemical reactions, and then always plot the null line. But of course you can do it now the other way around and say, hey, I want to have bistability. You see right away the bistability. I draw this line, like, okay, what chemical reactions actually, and there can be arbitrary many in general, reaction networks can lead to such a line. I think that's why I think I'm opening a new possibility to construct it, because I think going through all chemical reactions might be more tedious than doing the backward construction. And you can do with learning and other kind of things. But I think it's there. I think I would also constrain a little bit what is possible. I mean, I would not include a fifth order chemical reaction here. So I would go with classical bimolecular reactions in general and how to construct it.
Excuse me, here.
Here, yeah.
In the first diagram, so just checking if I understood correctly or not, the parallel lines, no, on the next board, the parallel lines between them is the homogeneous phase of the ternary mixture, and the stationary solution would fall in the on the tie line. Is that it? Maybe I didn't get it.
The parallel lines, these ones, these are two different conserved lines. And you can do arbitrary conserved lines. It's up to you how many molecules you add into the box at t equal to 0. And this sets a domain of two very special conserved lines that exactly intersect here in these fixed points. And in all conserved values, so each one corresponds to a psi value. So the range of psi values from here to here give you always phase separation. And all outside don't give you phase separation. I mean, this was here. The drawing here is a little bit bad, actually. It does not intersect again. It's just my hand drawing.
But the tie line would represent what exactly in the diagram?
Yeah. So this is a special tie line. Because it. So here, these are the range of psi values that lead to phase separation. No matter what psi value you pick, you always get the same composition inside and outside in the phase separated system. However, what is different along this line is simply the volume of phase 1 to 2. That's the only thing which you regulate.
Thank you.
And it's also intuitive, because you add more molecules to the box, the droplets are bigger. How am I doing in time? Yeah.
12 minutes.
Okay. Good. Obviously not my strength to be in time. Good. So now let's apply this to the system and see how far we go. You just interrupt me when I get a bit over time. So now we all understood these are the conserved lines, and then I'll show you the construction just for that system, because that's a system of interest, because only this phase diagram allows you for oscillations. Good. The average diff on that line, we have this conserved quantity. We have this reaction extent. The reaction is pretty nice, because the reaction extent exactly tells you where you are on this line. So the reaction just pushes the averages along this line, and it's exactly, when you put a parameterization on this line, it goes from minus to plus psi. So whatever value you put here, it's you go from here to here. So it's a parameterization of this chemical reaction along this line. Good. That's the line for that phase diagram. That is the chemical equilibrium line. It goes here. So it selects here this lower time line when mu A is equal to mu B. But this is not what we want to do. We want to fuel the system. Okay. So what reaction null line do we get? When we fuel the system, we get a bunch of different ones. So if the fueling amplitude is 0, then the band is not broken. We get the chemical equilibrium line. When we increase this one, we see something very interesting. So you see, like here, we get an intersection. Let's zoom in a bit. When we zoom in, that's how it looks like. We see the conservation line here intersects here with this homogeneous null clone outside. So we have one stationary state that's homogeneous and sits outside. It's essentially unfueled. But now if we introduce the fuel, the reaction null cline bends over step by step, and at some point it intersects with the conserved line, and then what you get is essentially exactly what I was illustrating also in my drawing on the board, is in addition to this fixed point, you get an unstable one, and here a stable one. Okay? So this we have explained here just with a different kind of drawing, and which this means we have bistability. So we can now illustrate the bistability slightly differently by doing a classical bifurcation diagram. So this is a phase diagram that looks very complicated. We can just use this equation to plot this on the y-axis and use the reaction extent on the x-axis. And then you see a diagram I think we're all used to, right? Have a look at this diagram. You plot here the average reaction rate, and here we go with the reaction extent along the conserved line. And then we see for enough fueling strength, the redder it is, we have exactly this intersection, and we see here a stable state here, and then here, this line here is exactly the domain, the binodal of phase separation. So we see we have a piecewise, here linear, and here by lowest order quadratic equation. It's a bit different to classical bistable systems, which usually have a third order polynomial, but this is just a distinct feature of this phase separation system. And then, because of the sake of time, I wanted to also maybe illustrate you a little bit how to calculate this. So actually, analytically, you can calculate now this right-hand side by just Taylor expanding around these values. And what you can see here, what is very important for the instability, so this one does not lead to this fixed point. So somehow this point here has to push through this axis. So the height here is very important. So you can now Taylor expand this, and what you get is what is in the field of linear dynamics and bifurcation theory called a normal form. A normal form is really the minimal mathematical structure that can give you this bistable, in that case, this bistable system. And the normal form has only this special feature here that it's piecewise. Yeah, it has a constant. This is exactly this offset. You can even calculate analytically, and this quadratic shape here. You see, like this is the leading order is a quadratic shape here. Yeah? There's some interesting math here because you have to connect the solutions. But I think for the sake of time and better show you some movies at the end, I skip this now. Okay, good. This is the corresponding bifurcation diagram, and you can recognize we have a stable solution all the time, and if you jump off this solution to a stable, opening up a stable and an unstable branch, and because it's upshifted, it's called a saddle node bifurcation in terms of the classical bifurcation. And also that's exactly what the normal form helps you to characterize the type of bifurcation. The interesting bit is when you can get the normal form, you can also calculate analytically the fuel strengths you have to exceed to get the bifurcation. It's very beautiful. Huh? Yeah, I mean, here on that slide I want to show you how that works, but I better skip this now to go a bit to the movies. Good. Bistability. This is not so bad. So we have two stable solutions. So now we have to either add fluctuations to jump from one solution to the next and make some Kramer's problem of two-state jumping process out of it. And we actually did this. And you can jump between stochastically between this and that state. And there's a barrier in between. You can also calculate analytically. But we want to go to oscillations. So what are we missing to get oscillations? Do you have an idea? So what would you add to the system, now from a science perspective, to get oscillations? I mean, you have seen, I mean, we got oscillations. So what would you add? Any idea? Well, this system is happy. It has two stationary states. But of course, the existence of a second one is it's maintained out of equilibrium. What do we need? We have to make it unhappy in some way. Yeah? I mean, it has two stable fixed points. We have to kind of kill the fixed points in some way. Yeah? What do you think? I mean, now it comes all these along one conserved line. We have these two fixed points. What would you construct? So you're a chemist. So what reaction would you maybe adding another reaction might be no possibility, but what type of reaction would you add? What's the job of that reaction? So you don't get it away from that line. What do you do then? You tell me how many molecules totally on the box, sets the conserved line. The system has these two safe fixed points, and then that's it. But, huh? Yeah, if you add noise, the system jumps from here to here, back and forth. Through it. If you add super crazy noise, you destroy everything, fine, but not enough. What type of reaction could we add? And I mean, the diagram tells it right away. So I showed this diagram to my chemist. I mean, he's really good. I think he becomes a theorist. And told me, like, yeah, I mean, we have to, A plus B is the conserved line. So we need a reaction that changes the conserved line. Okay, because then you have a chance either you move around the fixed points or you destabilize them. And this you can do. You can cook up hundreds of reactions, of course. But you have to, on the left-hand side, you need a reaction that is some thought of A plus B reacts to something else. And since we did a simple theory, I mean, he said, like, no, you can't do this, it reacts to a real component, we let it react to the solvent, which is a nonsense reaction, of course. But otherwise, I have to do a mixture with 4 components or 5 components. And we can do this as well. But then I lose this beautiful illustration of doing something in a phase diagram. No? So again, here, the chemists would disagree, of course. Like, yeah, essentially, you have to attack this conserved line with a second pathway. And even if we don't maintain this out of equilibrium, it was completely unnecessary. It's just a passive reaction. But because the first pathway is out of equilibrium, the system is still an out of equilibrium system. Good. That's how we do it. This you know. You would be able to write this down now, right? Because like, oh, equilibrium, that's a stoichiometric coefficient. So at equilibrium, the Kemp potential A plus B is equal to 2 times mu C. If that is equal, it's equilibrium. But it can't because the other pathway is out of equilibrium. So we have no more conserved quantity. So what do we get? We get this. We get, yeah, what is this? Actually, is this a movie? Oh yeah, it's a movie, yeah, exactly. So I pushed the system away, and then it did some cool stuff, and then it got stuck again, and stayed. What is this called? Yeah, excitability. I heard it, yeah, thanks. Very slowly, it comes back at some point if you have time. These different time scales in the problem. We are close. So still, the problem is we did not kill both fixed points. We killed one. Okay, and then we get an excitable system. We can push it away, and the system then dissolves the droplets and forms it again. So we're close. So what's the last recipe? You have to find what else you have to do to get to oscillations. Then you have to kill the last fixed point. And you see, like, this fixed point is very close, so the dashed line always means it's an unstable branch. So we have to somehow do some modification that we get into this unstable branch. Yeah. And for that, we can derive the normal form, and it tells us exactly how to do this. Now the normal form has the same thing as before, but it has now a non-trivial OD for the recently conserved quantities, normal conserved psi bar. Good. And we did this. We pushed it into this unstable domain. There are multiple ways to do this. Doesn't matter. These are just the principles. And when it looks like this, it's just one unstable fixed point left. So what the system can do, it can do this. It starts here. Wherever we start, let it start. Doesn't matter. It does not really matter. There's the phase space, when you zoom out, is a bit more complicated. And you should recognize, I mean, this is now a trajectory where I only illustrate the average quantities, okay? And the black line is the binodal. So tie lines go like this, right? So when you're somewhere inside here, you sit on a tie line in the fast diffusion theory I derived for you, and you have a phase-separated droplet. And as the averages go up here, the droplet composition is changing, and this change of droplet composition creates exactly this feedback loop, and the droplet here at this point dissolves again. Then you can, this is the drawing, then this is one pulse solution that keeps on going forever, which is the droplet volume, for example. Formation and dissolution. Yeah, please. Yeah. Oh, yeah.
I noticed that there is some change in the speed of some regions. Is that something about like it's stuck in a metastable state and the perturbation is not enough to take out, or?
No,
Something
because
else?
I mean, we of course we can play with the direction rate coefficients a little bit, yeah, but the entire idea of excited media, when you think about this, is exactly you always have different time scales, how things relax dynamically. But we have here something that's a bit harder to control is that the actual things, how fast things happen, is always a reaction rate coefficient times an exponential of a chem potential. And you're changing this one. Yeah? So then you have a weighting factor of the exponential of the chem potential that can alter the rates, how the trajectories will actually move. Yeah? And this can be easy then by a factor of 10 over 100. So we played a little bit with the reaction rate coefficients to have a movie that looks acceptable. You don't have to wait forever, but this is a general problem of real chemical systems that different relaxation directions come with completely different time scales. Yeah. Good, and then of course we have to, let's go back to how we started. We started with these, with the fields, yeah? And with the Canhida type equation, and we only explored all this also analytically at this fast diffusion limit. So we can just run also the Canhida type of equations and check whether it's do the same thing. And actually, I've not compared now the trajectories. The trajectories look also very similar when you compare the trajectories, but also for the same type of parameters, you of course have a bit more freedom. You know, you can put a diffusivity, but you have to put it such that it corresponds to this fast diffusion limit. Otherwise, of course, it's a bit different. Yeah? But qualitatively, we see the oscillations also in the continuum theory. So I now know exactly at the time where it should end. Do you give me 5 more minutes, or then I show you 2 more movies? Yeah? Yeah? As a quick thing, when you look at the center, so everyone who's interested, you see a little bit of a black dot. The black dot was a trick to make sure that only one droplet forms. And that was we added a nucleation potential. But if you don't do this, you see cooler stuff. You see oscillations of many droplets. Because they start talking to each other with this external field, and they try to synchronize. And anyone in pattern formation has seen exactly these kind of patterns. It gets even worse. So when you look into a phase diagram, you don't see only these synchronized oscillations. Now we put this into a rather thin thing. You see arrested states. You see states that I can get synchronized. So the anti-column, they all come and go together as a team. But you can also get traveling waves, yeah, as in classical pattern formation. Again, patterns are not unique. It's good that we see all this now for that system. But it's a really different system. These are condensates forming and dissolving regular time. And if you find also chaotic patterns, etc., in case you get interested. Good. I will not go too much into this because I'm over time. Thanks for giving me these extra few minutes. You can ask about lots of questions about synchronization. And then let's go right to the summary. What I've shown you today is we could excite a little bit about these active droplets, and there's a bunch of different phenomena, but it's better to devote to one more accurately. Active droplets with out-of-room chemical reactions can lead to bistability, excitability, and single droplet oscillations. And when you allow droplets, with this, the generic, it's true by default, many droplets to form at the same time, they start talking to each other and lead to collective and synchronous active droplet oscillations. Yeah? Good. Yeah, I would like to thank the team who were involved. In particular, this is work from Iva and Hidde, and there's the collaboration with Böckhofen was done with Advet, and we're happy with having Jupp Böckhofen as a chemist collaborator and having this interaction between theory and experiment, which is very inspiring how to do theory in general. Now I would like to thank you for listening. I would like to thank in particular the organizers. I can't see them all the time. It was really amazing. Thanks. Thanks for making this really a wonderful stay. And yeah, I'm also ready. I'm still here a little bit after lunch. So if you have questions, reach out to me. Thanks for listening. And yeah, I'm ready to take more questions.
Thank you for the lecture. I have more question about the synchronization dynamics. Can you talk more about the synchronization or
Yeah,
Just like give me a.
I mean, as for all the
idea.
synchronizing systems, there's a length scale because it relies on transport in space, on which you can actually synchronize. Yeah? Otherwise, you get synchronized patches that grow to a certain characteristic size and they talk to each other. Yeah? So my first naive hope was that you get kind of long-range ordered synchronous states, but yeah, when you think about it more carefully, they have to talk to each other, and how does this long-range interaction come from? I mean, we didn't see this. We could just confirm that. And talking to other scientists, it was also confirmed this has never been seen, actually. Yeah? Long-range synchronized states. So, but anyways, this is ongoing research. Yeah? So we just look at classical order parameters, correlation functions, and investigate this. But we see, we understand now what sets the characteristic length scale for synchronization. So if you want, we can talk about that. But we've not seen any sort of quasi or long-range order of that.
Thank you.
Welcome.
Thank you for the lecture, Professor. And could you tell me one more time, how did you get rid of the second fixed point? Because I didn't understand.
Exactly, yeah. So if you add another reaction, it's like adding another constraint. Yeah? You remove one fixed point, but one can still remain stable. Yeah? It can still be a monostable system. But then the phase space has now changed such that upon perturbation, the system is excitable. And then you know you're very close, because excitability is the prerequisite for the oscillations. And along this null line, is it shown here? Yeah, here it is. Yeah, exactly. Along this null line, you see, so there's one null line here, this one here. It's still monostable, that's excitable case. Yeah? Here it's stable, and here it becomes locally unstable. So I have to find our way to push this fixed point, it still remains, to make it an unstable fixed point, to destabilize it. And there are different ways to do this, and then once you push it there, actually it's very consistent. Here you see it very nicely. The fixed point sits now here, and that's now the oscillating system. You have a set of a few parameters. You can change the internal free energies, for example, of the chemicals. Yeah? That's the best way, because the phase diagram remains unaffected, and you only affect one of, I think what we did essentially is to change the internal free energies of the solvent, because the other chemical reaction also remains unaffected, so you have a very nice way of keeping the red line, keeping this line unaffected, but only altering that one, because that's the first reaction, that it doesn't involve the solvent, and but that one depends on the solvent, so by just changing the solvent, the phase diagram remains the same, the red line, so you only move down the green one. So that's a trick. But you can do it also differently. But then you change two things at the same time, you don't know whether you go in the right direction.
Okay, thank you.
More questions? If not, let's give Chris a big round of applause, and thank you for the 4 lectures.